let us discuss the structure of the DNA as described by the model of Watson and Crick way back in the 1950s. So the DNA is a double helix structure. So that means you have the nucleic acid. You have two strands of nucleic acids that are entwined with each other in a double helix formation. So the diameter is generally around 2 nanometers in diameter. So this is your DNA structure. And we have 3 to 4 nanometer length for each full twist of the double helix. And between each base pair, so that means each nucleic acid, we have, between each nucleotide on the nucleic acid chain, we have 0.34 nanometer of distance. So that's the general shape and length, or rather a description of the model. And then one very important property of this model, as a double helix structure, the strands are paired, or rather their bases are paired. That is actually this explains why Erwin Chargaff always finds that the number of adenine is always the same as the number of thymine, and the number of guanine is the same as the number of cytosine, because they are paired. That means your adenine is paired with thymine and your guanine is paired with your cytosine. So they are in this model of the DNA structure, that we have the two strands. So again, this is a double helix, but for us to see very clearly the base pairing, so in here, you have one strand that are running in parallel or anti-parallel directions. When we say anti-parallel, they are parallel with each other, but one strand they are in the opposite directions. They are parallel but in opposing directions. So that means we have one strand that runs 5' to 3', and another strand that runs 3' to 5'. So what does the 5 and the 3 mean? The 5 and the 3 are the positions on the ribose. Remember, this is the oxyribonucleic acid contains a ribose sugar, and the ribose sugars, the carbon in the ribose sugars are numbered. And the 5 and the 3 are the positions, the numbers on the ribose sugar. So one strand lies from the 5' to the 3' position, and the other is on the 3' to the 5'. So the base pairing occurs between adenine and thymine, and they are paired. How are they exactly paired? They are joined together. The nitrogen bases are joined together by hydrogen bonds. Hydrogen bond is relatively weaker. They are not considered chemical bond. They are more of interatomic or rather intermolecular forces. So hydrogen bonds is the strongest intermolecular force. But they are technically they are not like covalent, whereas there's a sharing of electrons. The hydrogen bond is the bond that occurs between the hydrogen atom and a lone pair of an electronegative atom. Now your hydrogen, we have two hydrogen bonds joining the thymine and the adenine bases, and we have 3 hydrogen bonds joining the guanine and the cytosine bases. So if looking at it this way, it looks like a ladder. So you have the backbone of the ladder, which is the sugar phosphate backbone. We call it the sugar phosphate because this is the sugar, this is actually the ribose sugar, and you have the phosphate group. Sugar phosphate, sugar phosphate, sugar phosphate. That is the backbone of your ladder. Basically, it's the backbone of the DNA, and the bases are like rungs of the ladder. So it's a twisted ladder. Some people describe it as a twisted ladder configuration. So the bases are arranged in such a way that they are aligned flatly. So as you can see here, they are flat. Okay, so they are aligned in a plane, in the same plane, or at least a similar plane with each other. So they're aligned flat. Okay, so that is basically the DNA structure as described by the Watson and Crick model. And the complementary bases. So again, the complementary base pairing is explained by your Chargaff's rule. Now, this complementary base pairing is actually, or rather, it explains how the DNA is transferred from one generation to another, and it also explains how the information is protected. So again, we have complementary pairing, adenine to thymine, guanine, and cytosine. So if you look at the illustration, you can see here that there's a specific shape between adenine and thymine, which is different from cytosine and guanine. Because the bases, or the pairing of the bases, for example, adenine can only pair with thymine for the DNA, and cytosine with guanine. So adenine cannot pair with guanine, nor can it pair with cytosine. So they won't fit unless it's with their proper base pair. Now, this manner allows us to know, so that basically, if we know the sequence of one strand of the DNA, we know the sequence of the next strand. So this is actually the key to the replication, how DNA is replicated, how we produce multiple copies of the DNA, and how it is passed down from parent to offspring. Now, the sequence of the base pairs encodes your genetic information. That is why the proportion of the base pairs, that means adenine is not equal to guanine. There is always a difference. There are always differences in the what do you call that? In the ratio of adenine and guanine in among different organisms. They are not the same because they have different DNA. Their DNA are unique. And because they are unique, they do not have the same adenine and guanine compositions. So basically, the sequence of the letters, the sequence of the 4 letters of the DNA is your information. It's like the binary code in computers. The computers process information using just 2 characters, which is the 0 and the 1. For the DNA, it processes information using the 4 letters. A, C, T, and G. So basically, that is your DNA. Now, let's look at the eukaryotic chromosome. So the DNA in a eukaryotic cell nucleus is organized as 1 or more chromosomes that differ in length and shape. So what is a chromosome? A chromosome is a structure that consists of DNA and its associated proteins. So they carry part or all of the cell's genetic information. So, during most of the cell's life, each chromosome consists of 1 DNA strand. So, for eukaryotic chromosomes, remember, for humans, for example, we have 23 pairs of chromosomes. So, that means 46 pieces of chromosomes. And each chromosome has 1 DNA strand. So, that means we have 46 DNA strands in our cells. So, when the cell prepares to divide, it duplicates all of its chromosomes so that both offspring receive a full set of your chromosome. So each duplicated chromosome has 2 strands, or the sister chromatids, attached to one another at the centromere. So that's basically how it forms the unique X shape that was observed under the microscope during cell division. So it consists of the 2 long filaments matching a characteristic X shape. So this is the normal chromosome as it is, it should be inside our cell when it's not replicating. And when our cells replicate, it forms the characteristic X shape of the chromosome. That means there's a duplication. So this is a duplicate of the other one. So this is observed during mitosis or during cell divisions. So you can do that. So the chromosome structure, so each filament consists of a coil of DNA wrapped around spools of proteins called histones. Because if you remember, if you look at the DNA, it's a very, very long molecule. So it's like a thread. So if you do not wind the thread properly, it will become entangled and it may cause breaks. If there's far too much strain on the strand, it may break. So you must have a proper way to store the DNA without breaking it up. So you have proteins called the histones. So it's like a spool that the DNA winds into. So the DNA molecules consist of 2 strands twisted in a DNA helix. So basically, this is how the chromosomes form from DNA and its proteins. So you have your DNA double helix here. So that's your DNA double helix. It's a double helical structure. So it is actually it's similar to a very, very long thread. So that thread is wind around these little beads. These are the histone proteins. So they wind twice around each histone bead. And each histone bead is arranged in what you call a coiled structure. So they form somewhat like a beaded bracelet or a beaded string. So this is the nucleosome. So if a nucleosome is a structure of the DNA and the histone, or rather a histone and the DNA that is wound around it. So that's your nucleosome. So you have your this nucleosome joined together to form what you call a fiber, which is basically somewhat like a beaded bracelet structure, like bracelet. So this fiber coils around, coils of your fibers. So they coil around until you form your chromosome. This is now your chromosome. So that is how your DNA forms into your chromosome. And from its shape, you can see that it's a very, very, very long structure. It's actually very, very long. So if you stretch a single piece of DNA, the length can reach around in terms of centimeters. Okay, so that is how long a DNA molecule is. So these are electrons, micrograph structure of your chromosome. So there is a duplicated chromosome. So you have the characteristic X shape. So one sister chromatid and a second sister chromatid. This is your fiber, the coiled nucleosome. So you can see here the grainy part of this fiber, that is your nucleosome. And this is an extended nucleosome. So these are the histone proteins, and the string is the DNA. So that is your structure of the DNA nucleic acid. The total number of chromosomes in a eukaryotic cell is the chromosome number, is characteristic of the species. So human body cells, for humans we have 46 chromosomes, or the 23 pairs. Two of each type of we have two of each types. Our chromosomes occur in pairs. So we have what you call a diploid number of chromosomes. So the karyotype shows how many chromosomes there are in an individual cell, and it can reveal major structural abnormalities. For example, Down syndrome has we have an extra chromosome for a Down syndrome. And we have the XXY chromosome, XXY syndrome, or most of the abnormalities brought about by differences in different than normal number of chromosomes are usually makes that organism sterile or unable to produce an offspring. Okay, so this is our karyotype. This is for a woman's karyotype, for a human. So these are the pairs of chromosomes. So how are they paired? They have similar shape, length, and overall structure. So I call it the chromosome of a woman because it has the X chromosome and no Y chromosome. So if it's a man, it has one X chromosome and one Y chromosome. So in a diploid organism, one chromosome in a pair is inherited from the mother and one from the father. All except one pair of the chromosomes are autosomes. When we say autosomes, these are chromosomes having the same length, shape, and centromere location. Now, the remaining pair of chromosomes is called the sex chromosome because they gives us the difference between males and females. So females has two X chromosomes, where human males have an X and a Y chromosome. That is for the humans. When we talk about frogs, it's a different matter. When we talk about birds, it's a different matter. So the cells basically we'll discuss the replication in the next video.